Today we'll have two things going on. First, an introduction to carbohydrates lecture, which will take the place, which is the pre-recorded lecture. I'm just doing it live. And then the one on glycolysis. I probably will not finish. In fact, I know I won't finish the lecture on glycolysis, and we'll push the rest of that off till Thursday, at which time I plan to cover finish glycolysis and discuss the TCA cycle. Are there any questions before I get started? I do want to, of course, you guys had an exam yesterday, and I have some preliminary results, but I'm not finished analyzing all the exam results. So I apologize for that delay. When I see you on Thursday, though, and probably by this evening, actually, I'll be able to give you your results on the exam with any necessary commentary to go along with that. Okay, all right then. So let's get started. Today I'd like to start with a discussion of carbohydrates. And let's go back to the beginning of this lecture. Okay, so an introduction to carbohydrates. And Dr. Sutherland, are you able to record now? Can you hear me, Dr. Sutherland?
Yes, it's being recorded.
Great. Thank you. Thank you, Dr. Sutherland, for recording the lecture. Okay, so an introduction to carbohydrates. Then the objectives are listed at the end of this lecture, and the sources used for it are chapter 11 of your textbook, and the following pages from the Devlin textbook, 7th edition, and also chapter 7 of Nelson and Cox's Lehninger Principles of Biochemistry, 7th edition. So what are carbohydrates? Okay, well, they're hydrates of carbon, and they all have the formula CH2O sub N, where N is the number of carbons. So that's the molecular formula that they have. And so an example is glucose, which has the molecular formula C6H12O6, or in other words, C.H2O sub 6, where N is 6, meaning that glucose has 6 carbons and it's a hexose. It's known as a hexose. What carbohydrates are aldehyde or ketone compounds with multiple hydroxyl groups. And we'll see plenty of examples of them in this lecture today. They have different biological roles. They store energy. So for example, in animals, glycogen stores energy. Glycogen is a glucose, a storage form of glucose in animals. And in in plants, we have starch, which plays the same role of storing energy. Carbohydrates make up part of the structure of deoxyribonucleic acid and ribonucleic acid, as you saw recently in the lectures on those topics. They're components of the bacterial cell walls and the extracellular matrix around mammalian tissues. They're sometimes linked to proteins or lipids, forming what are known as glycoconjugates. There are different kinds of carbohydrates. We have monosaccharides, which are the simplest sugars. Examples include glucose and fructose. Disaccharides, which are made up of 2 monosaccharide units linked together. Examples include maltose, sucrose, and lactose. And then, we have the oligosaccharides. These are made up of 3 to 20 units that are linked in either a linear or branched fashion, and linear or branched chains. And finally, we have polysaccharides. These are made up of hundreds of units that are linked in a repeating fashion, and they include glycogen, cellulose, and chitin, which is a type of polysaccharide found in arthropod exoskeletons. So let's look at the simplest monosaccharides, the trioses. So we see here two examples of a triose. On the left is glyceraldehyde. Notice it has 3 carbons. That makes it a triose. It also has an aldehyde functional group. So it's an aldotriose, okay, glyceraldehyde. And on the right, we have what's known as a ketotriose, dihydroxyacetone, okay? You can see the ketone group. If there were no OH groups, that would just be acetone. But with the 2 hydroxyl groups, its name becomes dihydroxyacetone. Now, if you look back at glyceraldehyde, what you'll see is that this carbon in the middle has 4 different groups attached to it. The aldehyde group, the hydroxyl group, a hydrogen atom, and then this CH2OH group. 4 different things. And as you learned with amino acids, when an atom has 4 different groups attached to it, that atom is said to be a chiral center, So, or a chiral carbon. And glyceraldehyde has such a chiral carbon. Therefore, it exists in 2 different so-called enantiomeric forms. The D, which stands for dextrorotary, and the L, which stands for levorotary. And these names are derived from the fact that if you have a solution with only the D enantiomer, that solution will rotate plane polarized light in a right-handed manner, or clockwise manner, okay? Whereas the L enantiomer in solution would rotate plane polarized light in the counterclockwise or left-handed direction. Now, of course, if you had a racemic mixture, right? Racemic mixture, which contains equal quantities or concentrations of the 2 different enantiomers, the net rotation of plane polarized light would be 0, right? Now we can look here at this figure on the upper right, and we can see that one enantiomer is a non-superimposable mirror image of the other one. So we have one of the enantiomers here, shown on the left in the figure, and we see its reflection. If you were to somehow try to take that reflection out of the mirror and try to superimpose it on the other one, they would not superimpose. So they're non-superimposable mirror images. This is characteristic of so-called enantiomers. 2 forms of a molecule that are non-superimposable mirror images of each other. Okay. Now, only the D enantiomer is found in sugars. Okay. So although both D and L can exist in nature, we only find the D in sugars. So let's take a look here. This upper figure shows a ball and stick model for the enantiomers. This middle pair of structures shows what are so-called Fisher projection formulas or Fisher projections. Okay. And what are Fisher projections? They are a 2-dimensional representation of something in 3 dimensions. Now down at the bottom, we have something called a perspective formula, or in other words, a wedge and dash. So we can see here that the OH and the hydrogen that's attached to the chiral carbon of glyceraldehyde project in front of the plane of the page or the screen, okay? Whereas the aldehyde, CHO, and the CH2OH groups project behind the plane of the screen or the page. right? So see the dashes there. So wedge means in front, dash means behind. And you can see how, you know, the D and the L are different, right? L has this OH here on the left, whereas D has that OH on the right. Now, Fisher projections, again, are two-dimensional representations of something in 3 dimensions. And so the horizontal lines, the horizontal bonds, if you will, are projecting in front of the page, and the vertical ones are projecting behind. So anytime you see something on the horizontal, that means it's projecting in front, and the vertical are projecting behind. Okay, so that's encoded in the two-dimensional representation. The other thing you'll notice is that in this carbon before the last carbon, okay, now in glyceraldehyde, there are only 3 carbons, right? So it's this middle one. But the one before the last carbon, notice the OH group is pointing to the right. That's what you find in the D isomer, okay? The D enantiomer. In the L, it's pointing to the left, okay? So you'll always, for the sugars seen in nature, the take-home message is you'll always see that, pretty much, see that OH pointing to the right there. There may be a few exceptions where you'll see some L's, but most of the sugars have the OH to the right in the D configuration. Are there any questions about this? All right, so how many stereoisomers are possible for a sugar that contains N chiral carbons? So N here is indicating the number of chiral carbons, okay? Now, what are stereoisomers, first of all? They're molecules whose atoms are connected in the same order, but differ in their spatial arrangement. Atoms are connected in the same order, but they differ in their spatial arrangement. Now, we can figure out how many stereoisomers are possible for a molecule with a certain number of chiral carbons, okay? That number is 2 to the N, where N is the number of chiral carbons. So, aldotrioses like glyceraldehyde, have 1 chiral carbon, and so they have 2 different stereoisomers. We looked at those, okay? That's the D and the L. Aldotetroses, those with 4 carbons, which have 2 chiral carbons, okay, can have 2 to the N, or 2 squared equals 4 different stereoisomers. Aldopentoses can have 8, aldohexoses 16, and aldoheptoses 32. Okay, now this number includes enantiomers, both D and L, and diastereomers. These are stereoisomers that are not mirror images. Okay, and ketoses would have half this number. Why is that? Does anybody want to guess why ketoses would have half the number that aldoses have?
Because they have fewer chiral carbons.
Yes, that's correct. When you have that ketone functional group there in the middle of the molecule, that knocks out one of the chiral carbons. So you have fewer, therefore fewer stereoisomers. Well, let's take a look at, for example, the aldohexoses here. These are sugar molecules with 6 carbons, and there are 16 possible stereoisomers. Now, this 16 includes L isomers and D isomers. And again, you really only find D in nature. So that really, the number that you see in nature is 8. We're going to take a look at that. You can also look, by the way, at figure 11 too, that describes this a little more in your textbook. Okay, so now let's unpack the meanings of these different terms, okay, to help us get a firmer grasp on what these different kinds of isomers are. So what is an isomer? What are isomers? Isomers are molecules that have the same molecular formula, but different structures. That's straightforward, okay? Among these, we can have so-called constitutional or structural isomers and stereoisomers, okay? Now, constitutional isomers differ in the order of attachment of the atoms, okay? Same molecular formula, different structures that differ in the order of attachment of the atoms. So examples of structural isomers are glyceraldehyde and dihydroxyacetone, where you see how the C double bond O is in a different position, right? We have a different connectivity, or the order of attachment is different of the atoms. Constitutional. Stereoisomers, in stereoisomers, the atoms are connected in the same order, but they differ in their spatial arrangement. They have different configurations. So we talked about enantiomers. They're a kind of stereoisomer that are non-superimposable mirror images. And we saw the D and the L glyceraldehyde as examples of that. Diastereomers are also stereoisomers. They're stereoisomers that are not mirror images of each other, okay? So this was an error in your textbook. There should have been a hydrogen there. Actually, this is from the, yeah, this is from your textbook, right? So what do we see here? We see D-glucose. It's a very common monosaccharide we're going to be discussing quite a bit. C6H12O6, and then we have D-altrose. Also C6H12O6. They're isomers of each other, okay? And they're stereoisomers. Atoms are all connected in the same order, but differ in spatial arrangement. So how do they differ between these two now? Well, on the second carbon, we see the OH is to the left in D-altrose, but to the right in D-glucose. That's one difference. Now let's look at the third carbon here. In D-altrose, that OH is to the right, but in D-glucose, it's to the left. Now the configurations about the other two, next two carbons are the same, and this last one is not chiral, last carbon. So that shows you how there's no way that these would be mirror images of each other, okay? And yet they have the same connectivity, the same spatial arrangement. I'm sorry, the same order of connection, but different spatial arrangements, especially about the second and third carbons. Now notice that the OH group is to the left, to the right in both of these. That's how you know it's D and not L. If it were L, the OH group would be to the left. All right, now, among diastereomers, you can have so-called epimers and anomers. Now, epimers are molecules that differ at only one of several asymmetric carbon atoms in the structure. D-glucose and D-mannose are examples of epimers. So mannose is an epimer of glucose, and glucose is an epimer of mannose. And you can see how they differ. On the second carbon, in glucose, the OH is to the right, but in mannose, it's to the left. Otherwise, they're the same. So they differ at that one position. Anomers are isomers that differ at a new asymmetric carbon atom that's formed upon ring closure. We're going to discuss this. We're going to discuss how glucose can form a ring structure in which the OH group on the penultimate carbon, that's the second to last one, forms a bond with carbon 1. And you get this heterocyclic ring called a pyranose ring. Well, when that bond forms, the OH group on carbon 1 can be either and by the way, this used to be the aldehyde, C double bond O, which now becomes an OH, right? That OH can be down in the down position relative to the ring with the CH2OH at the top here, or it can be up on the same side as that CH2OH. When it's on the same side, it's called beta. When it's on the opposite side of that CH2OH up here, it's called alpha. These two are called anomers. So we can talk about the alpha anomer and the beta anomer. And the other thing I'll briefly mention is that so-called mutarotation is possible, in which alpha is converted to beta, or beta is converted back to alpha. And that actually occurs through what's called the open chain form. So this is an example of the open chain form of glucose, right? And so to proceed from one to the other, say beta to alpha or alpha to beta, you'd have to go through the open chain form where the ring can close and trap the OH group either below or above the ring. Any questions so far?
I have a question.
Yeah.
So when differentiating between the diastereoisomer and the epimer, the difference is that the epimer is only different at one point, and then the other one is different at more than one point?
Good question. So actually, an epimer is a type of diastereomer. I've given an example of a diastereomer here. Okay, I've given an example. But these are also diastereomers, these epimers. So an epimer is a subclass of diastereomers. Okay, anomers are a subgroup of diastereomers. Does that help?
Yes, so an epimer would also be considered a diastereoisomer. Okay.
Correct, that is correct. That is correct. I've just given an example here. I could have given epimers as an example, but I wanted to save that because I wanted to illustrate a specific type of diastereomer called an epimer. Does that help?
Yes.
Good question, thank you. All right. Now.
Excuse me.
Yeah.
I have a question from the last slide.
Oh, yeah. This one here?
No, from right here. Yes.
Yeah.
So just for clarity, to tell if it's D or L, we're looking at the chiral center.
What you're looking at is that OH that's attached to the penultimate carbon, which is the second to last one. So this carbon of the CH2OH is the last carbon. That's called the ultimate. The one above it is called the penultimate. Notice how the OH group goes to the right in both of these. That means that's why they're both D, okay?
Okay.
Well, let's look at glyceraldehyde over here again, okay? See how in the D it's to the right, but in the L it's to the left?
Yes.
Now, I will say this. This assumes that you are displaying these molecules in a particular manner. So in all of these, say, aldoses, right? The aldehyde group is at the top. See that? Carbon 1 is the carbon of the aldehyde in these aldoses, okay? And then you go down from there. And that follows the convention of the Fisher diagram, where the aldehyde is at the top and the CH2OH is at the bottom, and that's the orientation. Of course, if you flipped it, everything would be different, right? So you have to have a certain orientation to talk about the OH group on the penultimate carbon being to the right or to the left. Does that help?
Yes, thank you.
Okay, you're welcome. All right, now, aldoses versus ketosis. So we talked about the difference, right? So in part A, you see D-aldose is up here. In part B of this figure, which is from the Leninger textbook, 7th edition, you see D-ketosis. All right, let's take a look at those. So this would be an aldotriose. We talked about it, D-glyceraldehyde. Okay, and then, but what about aldotetroses? There's a common one called D-erythrose. Okay, there's another one called D-threose, but D-threose doesn't occur in nature, and so the boxed names are the naturally occurring sugars that we're going to look at, we're going to consider in our discussions in this course. Okay. Again, ketoses have one fewer chiral carbons than the corresponding aldose. Ketoses have the ketone functional group. Aldoses have the aldehyde functional group. Now let's look at so-called aldopentoses. For that, we have D-ribose. Okay. D-arabinose and D-xylose. As the common naturally occurring ones, there's also D-lixose. And we can look at the differences among these. Look at the position of the OH group, right? So in D-ribose, it's to the right on the second carbon, but in D-arabinose, it's to the left. Somebody tell me how D-ribose and D-arabinose are related to each other. How are they related?
They're epimers.
Yes. All right. Here are the 8D hexoses, right? 8 is half of 16. These are the Ds, okay? We have all of these different ones here. D-glucose, D-mannose, and D-galactose are the ones that we're going to consider in the course. In terms of ketoses, we have dihydroxyacetone. We talked about that, the simplest ketose. We also have D-erythrulose, okay, which, as you can see, has this one chiral carbon here. And then we have ketopentoses, D-ribulose, D-xylulose, and then in terms of the ketohexoses, D-fructose. This D-ribulose and D-xylulose and D-ribose, we will see again when we consider what's called the pentose phosphate pathway later in the course. Are there any questions so far?
Yeah, I have a question.
Yeah.
Are you going to give us the structure and then maybe ask us a question if it's enantiomers or diastereomers, or you just want us to know by the box ones?
Yeah, I will. Well, if it's a common one that we've discussed, I might just ask you. There's certain ones we're going to discuss more than others, and for those you should know. You will know if you've studied. Okay, let's go to the next one. So here's an example, okay? So we can look at some, I'm going to move this up here, this little bar here. Some aldopentoses, okay? D-ribose and deoxyribose, right? Now you've seen these when you saw RNA and DNA, because we know that D-ribose is part of the structure of a ribonucleotide, and D-deoxyribose is part of the structure of a deoxyribonucleotide. And you can see that on the second carbon, which would be the 2' carbon in the ring form, right? Instead of this OH group, there's just simply a hydrogen atom in deoxy. You know that already, right? So, all right. So it's deoxy is a modified sugar that's found in DNA. Now, reiterating what epimers are, glucose and mannose are epimers of each other, and glucose and galactose are epimers of each other, but mannose and galactose are not epimers of each other. They're diastereomers, but not the specific kind of diastereomer called an epimer. Now, we already looked at glucose and mannose, didn't we? But let's look at glucose and galactose. How are they different? Well, go down the structure, we see that on the 1, 2, 3, 4, on the fourth carbon here, whereas the OH is to the right here on glucose, is to the left on galactose. And what we'll see is when these different sugars form their pyranose rings, okay, in glucose, that OH on the fourth carbon is going to be pointing down. And whereas the one on the fourth carbon in galactose is going to be pointing up. And that's a distinguishing feature between glucose and galactose. Now, fructose, right, is a ketohexose. It has 6 carbons, 1, 2, 3, 4, 5, 6, but it also has that ketone functional group. And when it cyclizes, it also will cyclize, it will form a furanose ring. And we're going to see that in just a minute. Any questions? Okay. Now, before I talk about cyclization of sugars, I need to give a small review, a brief review of some organic chemistry and discuss what are called hemiacetals, hemiketals, acetals and ketones. So when an alcohol reacts with an aldehyde, it forms what's called a hemiacetal. Okay, you can see here that the oxygen of the OH has attacked the carbonyl carbon, right? And one of the hydrogen atoms, that hydrogen atom has now gone to this oxygen up here to form an OH group. And so this is called a hemiacetal. Okay. When another alcohol now reacts with a hemiacetal, it forms what's called an acetal. An acetal and water. Okay. Okay. Now, when an alcohol reacts with a ketone, it forms what's called a hemiketal. So how's it different? Hemiketal has an R group here rather than a hydrogen of the hemiacetal. And then when another alcohol reacts, it forms a ketal now and water. Okay. So that's a little bit of review there. And we're going to see how this is relevant in just a minute. So when glucose forms a cyclic form, when it cyclizes, it forms what's called a hemiacetal. Okay, and let's look at that cyclization of glucose. It involves an intramolecular nucleophilic attack of the penultimate OH group on the carbonyl C1 carbon. Okay, and again, a hemiacetal is produced. So let's take a look at that. Here we have glucose. Okay, we're going to lay it down on its side such that the aldehyde group is to the right, and the aldehyde is to the right, and the CH2OH on the 6 and the 6 carbon is to the left, and then we're going to curl that around, such that the penultimate, the OH on the penultimate carbon is positioned to attack the carbon of the carbonyl of the aldehyde. When it does that, it forms a ring. Note the error in the ring of structure here. There should not be a hydrogen attached. That's a mistake. Okay, it's an error. But the C double bond O now becomes COH, and that COH can be either up to form beta or down to form alpha. Okay. Now, actually, beta is up only because this CH2OH is up, okay, which is the case for D, okay, which is the case for the D isomer. When there's a D isomer, the CH2OH is in the up position, okay, and the beta form, which has the OH on the same side as the COH, is in the up position. In the alpha, the OH on carbon 1 is down relative to, and on the opposite side of CH2OH. Okay. And this one's called, on the right, beta D glucopyranose, and on the right, alpha D glucopyranose. That's sort of the full name, if you will. Pyranose refers to the type of ring that's forming. Now, you can convert alpha to beta and beta to alpha, as I mentioned earlier, through a process called mutarotation, which proceeds through this open chain form. So the ring can open, and then the OH group can end up being on the other side of the ring. And there's actually, when you consider glucose in solution, there's an equilibrium distribution of these different forms, such that 33% is alpha, the alpha anomer, 66% is the beta anomer, and 1% is the open chain form. Okay, now we're going to consider fructose in a minute, but does anybody have any questions before I move on?
Yeah, I have.
Dr. Burns?
Yeah. A couple of questions. Yeah, go ahead.
You can go, Ian.
Go ahead, Jordan.
Is
is the note in the error ring. Where is that error at? You're saying that pay attention to the hydrogen, but it looks like when it does form the alpha, the hydrogen's still there. So where are we noting that error at exactly?
Yeah, this hydrogen on the oxygen in the ring. That should not be a hydrogen on that oxygen.
Okay, thank you.
Yeah. Mhm. If there were, there'd be a positive charge there, but it leaves and goes to the oxygen, the C double bond O, to form an OH. Actually, it leaves and goes into solution, and then another hydrogen can join. You'll see in a minute. So you see here, you see how there's no hydrogen attached here? Okay. See that? This is correct. So now, I kept talking about something being above and below the ring. And that's because we can represent the ring or think of the ring as being three-dimensional. And actually, when you draw a ring like this, you draw it in such a way as to make it perpendicular to the plane of the page or the screen. So this is called a Haworth perspective or Haworth projection. And you can see that, you know, this part of the ring is pointing out toward you. That's why it's fatter, to show it's closer up. Whereas the back of the ring is pointing behind the screen. Okay, so this is thought to be perpendicular with the plane, and different groups are above and below the ring. See that? So in alpha, right, for the D isomer, this CH2OH is pointing up, and the OH is pointing down on the opposite side of the ring. Okay, for the beta, that OH, of course, would be up there, on the same side as the CH2OH. This is called a Haworth perspective or projection, and this is the open chain form, which is shown as a Fisher projection. All right, now let's turn to fructose and see how it cyclizes. So here, this is the structure of fructose. Let's lay it down on its side, curl that around so that the OH on the penultimate carbon can attack. CH2OH is pointing up here. It attacks and it forms this ring. This, and then you know, the OH can be up or down. The CH2OH can be down or up. Okay, so again, you can have alpha and beta anomers for D-fructose as well. This is called D-fructofuranose, because the ring, that five-membered ring, is called the furanose ring. Okay, and it's a hemiketal. It's a hemiketal. So how is it a hemiketal? Let's take a look. So this OH is an alcohol. It is attached to the same molecule, right? So it's an intramolecular hemiketal that forms. And this is an intramolecular nucleophilic attack. It attacks there. So here's the alcohol attacking the carbon, right? And you have a carbon with an OR, right? An R, an R, and an OH. Let's go back real quick. Right? Carbon with an OR, an R, an R, and an OH. Hemiketal. The hemiacetal right here for glucose is carbon, OR, R, OH, H, and OH. So let me say that again. Carbon, OR, R, H, and OH. Let's go back. Carbon, OR, R, H, and OH. So you can see that that was a hemiacetal for glucose. Hemiacetal for glucose, hemiketal for fructose. What else do I want to say? The other thing I want to say is that even though, so I've represented, you know, Haworth projections, right? Haworth projections or perspectives that show the ring being perpendicular. But it also shows the ring being flat, okay? Like this one here, as if it's a flat structure, albeit perpendicular to the page. But in fact, that's not the case, okay? The ring of glucose, for example, this is glucose, can in fact exist in two different conformations, in something called the chair conformation and another called the boat conformation, okay? Now, the chair, these two forms are not energetically equivalent, okay? The chair form is more stable and is preferred. And the reason is because the larger groups around the ring are in so-called equatorial positions. So see the E's here? That means equatorial. That means pointing out away from the ring as opposed to above and below the ring, okay? Now, so you see the CH2OH is in equatorial position. This OH is, that one is, all of them, all the big groups. Whereas what are in the axial positions? Hydrogens. See that? Hydrogens. Now, let's look at the boat form, though. In the boat form, it's different. Okay, when glucose forms a boat form, or goes into the boat form, you can see how, look at this OH, and that OH is up there. We've got some different larger groups now in axial positions where there can be steric clash, steric hindrance, right? That's why this is less preferred. Boat is energetically less preferred than chair. Both can form. It can exist in both, but certainly the equilibrium distribution would be skewed toward the chair form, okay? Now I'm going to ask you guys a quick question. What's the difference between a conformation and a configuration? Can anybody tell me that?
Is the conformation the ring structure and the configuration are how the atoms are in the ring structure?
I think you're on the right track. In a conformation, to go from one to the other, no covalent bonds would have to be broken. Let me say it again. To change conformation, no covalent bonds have to be broken. To change configuration, they do. So if you were to try to say change D to L or L to D, you'd actually have to yank off some atoms and attach them on the other side, right? One can't flip to the other spontaneously without breaking covalent bonds. Whereas in conformations, they can, okay? So that's the whole thing about conformations. And you'll later on, and in fact, we already have talked about a protein changing conformation, haven't we? Like hemoglobin going from the T to the R state or the R to the T state. Those are two different conformations in which the hemoglobin molecule tetramer can exist. No covalent bonds are broken to go from one to the other, right? So that's analogous. Now, the last thing I want to point out here is that just like this pyranose ring of glucose can exist in different conformations, the furanose ring of fructose can too, okay? And so the D-ribose of DNA and RNA can exist in so-called C3 endo and C2 endo envelope conformations. To learn more about those, you can look at your, read your textbook.
Can you repeat what you said about, you said in conformations, no covalent bonds must be broken, but in configurations, they're allowed to be broken?
They have to be broken. So let's go take a quick look, all right? We're gonna have to go way back now. Let's go back here. Let's look at glyceraldehyde, okay? And let's look here. This is D, that's L. These are two different configurations for glyceraldehyde. See how the OH group is on the right for D and on the left here in this Fisher diagram for the L? To get it from one to the other, you'd have to actually break that bond, that CO bond, and move it, right? You have to break that CH bond and move the atom. See that?
Yes.
The different configurations.
Okay.
All right. But for here, oh, look, just the molecule can just flop over and form this other conformation. Okay. All right.
Dr. Burns?
Yeah.
Dr. Burns, when you're saying the furanose ring can be the same thing, you mean it can also be in chair form and boat form?
Yeah. Oh, The
Okay.
furanose. No,
Yeah.
the conformations for a furanose ring are called the C2 endo and the C3 endo envelope conformations. It's like a puckering of the ring where either the C2 or the C3 is in the up position, sort of like the, you know how when you have an envelope, you take the top and you fold it down, or you can fold it up, right? That part that can stick up that you fold down. That's why it's called envelope. If you look at the figure in the textbook, it will become more clear.
Okay, thank you.
Yeah. Okay, so we can talk about a sugar being a reducing sugar or not. Okay, so let's unpack what that means. All reducing sugars have or can form a free aldehyde group. Okay, a common test for reducing sugar using what's called Fehling's reagent, F-E-H-L-I-N-G'(S), Fehling's reagent. It employs an alkaline copper 2 solution. And when a reducing sugar is present, a red brick precipitate will form. And down here we see what happens. But examples of reducing sugars include glucose, galactose, mannose, fructose, maltose, lactose. So you can have disaccharides that are reducing sugars and monosaccharides, okay? Let's take a look at what happens in this reaction that takes place for a reducing sugar. So glucose is a reducing sugar, it's shown here. It of course can form the open chain form spontaneously. About 1% of the glucose in solution is in the open chain form, which has a free aldehyde group. That group can be oxidized to a carboxylic acid. So there's an oxidation, and there's also a corresponding reduction reaction that occurs where copper 2 is reduced to copper 1. And under alkaline conditions, it forms this copper 1 oxide, which is brick red in color and insoluble. So it forms a brick red precipitate. Okay. And so you get a positive test. Now I'm going to ask some questions here. First question I'm going to ask is this. And you get a very robust, strong reaction when you have glucose. But here's my question. If the open chain form constitutes only 1% of the equilibrium mixture of glucose in solution, why would you get a robust result? Lots of brick red precipitate. Why? I know somebody knows.
Can you repeat the question?
If the open chain form, which has the free aldehyde group, comprises only 1% of the equilibrium mixture, right? The other 66% alpha, 66%, 3% beta, they don't have a free aldehyde, only the open chain form does. Why do you nonetheless get a very strong result with lots of brick red precipitate?
Because you have the open aldehyde, the open chain with the aldehyde in it.
Right. You have that open chain, that aldehyde form, and it's only 1% of the equilibrium mixture. Why does all the sugar react? Why does all of it react? In the sample.
Yeah, I think like when this form of sugar reduced and this equation will move to the right, like to produce more free aldehyde group, this form of sugar. So it will like keep the reaction like moving.
That's correct.
Yeah.
That's a manifestation of what's called Le Chatelier's principle. Right? As you use up this form, the equilibrium continues to shift and shift and shift until you've used all of it up. It's because these different forms are in equilibrium. That's called the Chatelier's principle, and we saw it when we were looking at, exhalation of oxygen. I'm sorry, inhalation of oxygen, exhalation of CO2, right? Where, as CO2 is exhaled, it pulls on a series of equilibria involving the red blood cell and hemoglobin and blood, right? Causing even more to be exhaled. And all of these, because all these equilibria are connected to each other. Same idea here. Now, what about fructose? Why does fructose give a positive test? I say it gives a positive test. Here, it's a reducing sugar. Why is it a reducing sugar when it has a ketone, not an aldehyde in its structure? And the answer is because fructose can undergo what's called a ketoenol tautomerization process, where the ketone is first converted to an enediol intermediate, then to an aldehyde. And that aldehyde group can then react, right? Now, why not sucrose? Why is sucrose not a reducing sugar? And the answer is because its C2 carbon is tied up in forming an acetal linkage, and so is not free to form a free aldehyde. We're going to take a look at that. Excuse me. We're going to take a look at that in just a minute when we look at the disaccharides. Folks, I'll probably go directly to the hour and then we'll take a 5-minute break, okay, at which the recording can just be paused until we continue. Let's look at some modified sugars. These are sugars that have some group attached, okay, some functional group. So we can consider what's called the glucose family, shown in this blue highlighted area. The amino sugars, these are sugars that have amino groups attached, shown in the red part here, okay, so-called deoxy sugars. And then acidic in this box, right? So these amino sugars are, right. And then we can consider acidic sugars. So let's take a look at some of these. So glucose is not a modified sugar, okay, right? But glucose 6-phosphate is. So it has a phosphate attached to the oxygen on the 6 carbon, right? Glucose 6, beta D glucose 6-phosphate, all right? Look at these here in the middle, beta D glucosamine, where there's an amino group instead of an OH group on the 2nd carbon, right? And then we can have N-acetyl beta D glucosamine, where there's an acetyl group attached to the nitrogen of glucosamine. That N just means that it's an N-linked, that the acetyl group is attached to the nitrogen. So N-acetyl beta D glucosamine. By the way, we sometimes refer to this as what's called glicnac, okay? And then we have also muramic acid, which is related to glucosamine, but also has this extra R group, which is shown here. That's what that looks like. And then N-acetyl muramic acid, where we have an acetyl group attached to the nitrogen of muramic acid. By the way, we see muramic acid in the bacterial cell wall structure, as we'll see later. Now, so galactose can also be modified with an amino group to form beta D galactosamine. Mannose can too, beta D manosamine. Okay. We talked about some deoxy sugars, D-ribose, but here's some others. Beta L fucose and alpha L rhamnose. So these are L sugars, so they're different. And notice what they are. Let's count the number of carbons, okay? 1, 2, 3, 4, 5, 6. But instead of having a CH2OH, you've got a CH3 there. So instead of having the OH here, right, CH2OH, you've got an extra hydrogen in place of the OH, so it's CH3. That's why it's deoxy. It lacks that OH group on the 6th carbon. Now, how is rhamnose different from fucose? Well, that OH is in a different position. This one is. That was the same. That was in a different position. So it's a different. They're different from each other. They're diastereomers, aren't they? Looks like they are. Same molecular formula. Diastereomers, different arrangements of groups, different configurations. Now I want to point something else out here. Okay. Notice that this CH3, sorry, points down. That's how you know it's L. Okay. It's down here also for rhamnose. That's how you know it's L. Now notice here that the OH group of beta-L fucose is on the same side as the CH3. That's why it's beta. But here, the OH group in alpha-L rhamnose is on a different side from the CH3. Therefore, it's alpha. Okay? So those are some nuances that when you understand them will help you to understand what alpha and beta mean, what L and D mean. Okay. And then finally, looking down here, we can consider some acidic sugars. And we have beta-D glucuronate, beta-D glucuronate. It's just a derivative of glucose in which there's, instead of a CH2OH, there's a carboxylate group there. And then we have D-gluconate, okay, in which basically aldehyde has been oxidized to a carboxylic acid group. Now we will see this later when we look at the pentose phosphate pathway, and this other one too, D-glucono-delta lactone, in which the aldehyde, in which the this carbon now has been oxidized to what's called a lactone, right? And then finally, on the right here, we have N-acetyl neuraminic acid, which is an example of a sialic acid, where you can see, you know, there's a number of modifications. In this structure. And R is this group right here, so that R is that. Are there any questions? Okay. The disaccharides, we're going to talk about that. This is really a pretty long lecture here, folks. We're just going to go ahead. Let's take a break now. Come back in 5 minutes, and we'll just continue this lecture. Okay. Dr. Sutherland, I'm going to get started again. You can restart the recording.
Okay, it's recording. Okay.
Thank you very much. Okay, so now I'd like to talk about what are known as disaccharides. So these are sugars that contain 2 monosaccharide units connected by what's called an O-glycosidic linkage. And for a molecule like maltose, which is an acetal, that's called an acetal linkage, okay? And these disaccharides are either acetals or ketals. So I suppose that for a disaccharide like sucrose, that linkage would be called a ketal linkage. Whereas for maltose, it's an acetal linkage. We'll look at those, okay? So examples of disaccharides include maltose, which is made up of 2 glucose units that are connected by a so-called alpha-1,4 O-glycosidic linkage. Lactose, which has a galactose and a glucose connected by a beta-1,4 O-glycosidic linkage. And sucrose, which contains a glucose and a fructose connected by a so-called alpha-1,2 O-glycosidic linkage. We'll look at each one of these, okay? That O-glycosidic bond can be enzymatically hydrolyzed, releasing the monosaccharide sugars. So maltose is cleaved by the enzyme maltase, sucrose by sucrase, and lactose by lactase, which is also known as beta-galactosidase. Now, many of these disaccharidases, if you will, the ones that hydrolyze that linkage between the two sugar units, are actually located on the in the intestinal lumen, in the epithelial cells that line the intestinal lumen. So they are located there, such as sucrase, lactase. Now, although infants are lactose tolerant,
Dr. Burns?
Yeah.
Did you say disaccharide and another name for the enzymes? Did you say something about disaccharide like A's?
yeah, I just called it a disaccharidase, or
Okay,
just
thank you.
an enzyme that will cleave, hydrolyze disaccharide. Infants are lactose tolerant, but many adults are lactose intolerant. That is, they can't digest lactose. And the reason is because they lack the enzyme lactase. They have a deficiency in the expression of the gene for the enzyme lactase, which is what's needed to break down lactose. And if you are lactose intolerant and you drink milk, you'll experience gas, bloating, diarrhea, and abdominal cramping. And turns out there are at least two separate evolutionary origins of what's known as lactase persistence, which is the persistence of lactase activity into adulthood. One of those origins is in Western Europe, and the other is in Africa. And both are related to the dairying industry. In other words, it arose in populations that had cows and carried out dairying and depended on cow's milk. And it's fascinating. In your objectives, you will see there an objective asking you to look up a scientist by the name of Sarah Tishkoff at the University of Pennsylvania. She's done research in tracing the African origins of lactase persistence. And there's actually more than one. And those genetic mutations that allow lactase persistence are different from each other and different from the one in Europe. So which means that they arose separately. And this is an example of what's called convergent evolution. Convergent evolution, where different, with the same phenotype, right? Lactose tolerance arose in completely different populations. It converged, if you will, on the same phenotype, even though their origins evolutionarily are different. That's called convergent evolution. Okay, so let's take a look. Is there anything else I need to say there? Let's take a look at some disaccharides. Maltose. It has an alpha-1,4-O glycosidic linkage. So here's one glucose unit, alpha D glucose, and here's another, beta D glucose. Here's the hemiacetal, right? Here's an alcohol, if you will, right, with its OH group. That's going to attack the carbon, which can form an aldehyde, right, because that ring can open up. And a condensation reaction will occur. Water will be eliminated, and we will form this disaccharide with an alpha-1,4-O glycosidic linkage. What does that mean? Well, the linkage is between carbon 1 of the sugar on the left and carbon 4 of the sugar of the glucose on the right. So it's called 1,4, shown here, okay? And this OH was in the alpha position. And so it's alpha 1,4. Now, this OH on the glucose on the right at position 4 is always down. It can't be up or down, right? You can't have two different anomeric forms in relation to the fourth carbon. Only the first one, right? So it's an alpha 1,4 O-glycosidic linkage, or using a really long, complicated name, alpha D glucopyranosyl 1,4-D glucopyranose. That's sort of like the full name, if you will. Now notice that this carbon on the sugar unit on the right, this carbon 1, is an anomeric carbon. It can open up, and you can have both beta as shown here, or alpha. So both alpha and beta anomers exist for maltose. That's why it's a reducing sugar. All right, we're going to take a look also at lactose and sucrose, and also trehalose is shown here as well. Let's look at lactose. Let's look at lactose. So you have, see the OH on the fourth carbon is up. That means this is galactose. Galactose. And over here, the OH was down. That's why it was glucose. And notice that this OH was up and formed a linkage. And so it's called beta. Beta 1,4. It's a beta 1,4 O-glycosidic linkage. Different ways of representing it. Okay. This OH of lactose can be up in the beta configuration or down. That's so. Lactose is also a reducing sugar. This ring can open up. You can form a free aldehyde. It can react. It's a reducing sugar. And this is the full name. Beta D galactopyranosyl 1,4 beta D glucopyranose. Okay, you look at these, a few of these, you'll see how they're coming up with the names that they came up with. Okay, now look at this in the middle. In the middle, we have what's called sucrose. It's a disaccharide. It's table sugar, which you put in your tea and coffee, produced by plants. Sugar beet, cane, sugar cane. You can see here you have a glucose, and then over here a fructose. And notice that it's this carbon here, carbon 2 of what used to be the anomeric carbon is now tied up in forming this O-glycosidic linkage, right? So it's an alpha 1,2 linkage. Alpha 1,2. And now, sucrose is not a reducing sugar, okay? Because this carbon, which would have been the anomeric carbon, is tied up in forming this bond, this linkage here. Is that an acetal linkage, or should I have said a ketal linkage? But at any rate, it is an O-glycosidic linkage, okay? So now this carbon over here, carbon 5, is not an anomeric carbon. The ring can open up, and so you cannot. This will not reduce Fehling's reagent. It's not a reducing sugar. Notice that this second ring, the fructose, is flipped, right? So you flipped over the ring, and then the bond was formed here. Now, trehalose, I'm going to show you trehalose just to illustrate that it's also not a reducing sugar. So there are two glucose units. Okay, the second glucose is flipped, and carbon 1 is forming, participating in the O-glycosidic bond. This bond is coming from the alpha position on the first glucose. So it's an alpha 1,1. Alpha 1,1 O-glycosidic linkage in trehalose, which again is not a reducing sugar, just like sucrose is not. Now, trehalose actually is used, I believe, as a sweetening agent. But you know, I read something recently rather disturbing about trehalose, which is that some bacteria had developed the ability to degrade it, and is associated with the aberrant maldistribution of gut bacteria. That when people consume trehalose and they happen to have a certain strain of a bacteria that's not good, its population just simply explodes because it can metabolize this sweetening agent, and that causes dysbiosis, problems in the gut. Reading about that. Anyway, so, but that's what trehalose is. Are there any questions?
Yes, question. Do you want us to know how to name this, the structures?
How to name? Yes, you need to be able to understand. I'd say understand more than name, because I'm not gonna give you a structure and that you haven't seen before, and say name this. But I might give you a structure that you have seen before, like one of these, and ask you to tell me what it is and what kind of linkage is present. Does that make sense?
Yes, thank you.
Okay, you're welcome. So now let's talk about polysaccharides. Okay, there are different kinds of polysaccharides. One of them is starch. This is a storage form of glucose in plants. It has two components: amylose, which is a linear polymer of glucose. It's essentially polymaltose and has only alpha-1,4 linkages that are like the kind seen in maltose, right? And the other component of starch is amylopectin. This is branch now, not linear, a branch polymer, and it contains so-called alpha-1,6 branch points, as well as the alpha-1,4 linkages, and the branch points occur every approximately 30 residues. Glycogen is a storage form of glucose in animals. It's similar to amylopectin, but the branches occur more frequently, every approximately 10 glucose units, and it's a way of storing energy for quick release or to maintain blood sugar levels. So you have a muscle, a form of glycogen, and a liver. Muscle is used for quick energy during vigorous exercise, and the liver form is used to store glucose so that it can be released when needed from the liver, according to the blood sugar level that exists. Now, cellulose is a linear polymer of glucose units that are connected by beta-1,4 linkages. Alternating glucose residues are flipped. So the strands are straight, they're linear. If you look at glycogen or starch, and if you look at, for example, amylose is a good example, where you have alpha-1,4 linkages, the polymers tend to form these whorl-like structures, W-H-O-R-L, whorl, sort of a spiral or whorl-like structure. Whereas in cellulose, because every other residue is flipped, you don't see that, and the strands are linear. This allows them to form fibers, where the strands, individual strands of the fiber are connected by hydrogen bonds. Cellulose fibers have high tensile strength. They're found in the woody stems of plants and blades of grass. They're hydrolyzed by the enzyme cellulase, which humans lack. That's why we can't eat wood, basically, or grass, although we do need fiber in our diet. Termites, ruminants, cows, so on, have an enzyme, the enzyme cellulase, by virtue of specific symbiotic bacteria that are present in their guts. And finally, glycosaminoglycans are examples of heteropolysaccharides. So let's take a look at starch first. It has those two components, amylose and amylopectin. Shown in A is amylose. You see the sugar units all connected by alpha-1,4 linkages, okay? We can talk about the chain of amylose residues. We can talk about amylose, which exists as a chain of glucose units, as having a so-called reducing end, right, and a non-reducing end. Okay, and we'll see, yeah, okay, and so I just want to point that out. You can also look at amylopectin, and in amylopectin, what you have are these alpha-1,6 branch points where, you know, there's an O-glycosidic linkage between carbon 1 and carbon 6, forming a branch point. And so if you look at the structure of starch shown here in C, you can see, you know, amylose linear polymer interwoven with a one of the branches of amylopectin, which shown here has many or several of these alpha-1,6 branches, right? Coming off. Okay? It's highly branched. Now, the amylose molecules will have reducing ends, but the amylopectin, which is branched, will have only one reducing end, okay? And you'll have many non-reducing ends, okay? That's starch. I want to show you glycogen in just a minute because it's, do I talk about glycogen? Not as much as I would like. I'm going to talk about glycogen, of course, much, much more later. So let's look now a bit more closely at starch and glycogen. Because of the alpha-1,4 linkages among between glucose units, again, it has this curvature and forms a whorl-like structure. Whereas in cellulose, because every second residue is flipped relative to the one before it, the chains are straight and they are connected by these intra and inter chain hydrogen bonds that gives cellulose a very high tensile strength. And of course, in cellulose, the glucose units are connected by beta-1,4 linkages, beta-1,4. And you know, it's the beta-1,4 linkage that we can't hydrolyze. We don't have the enzyme to hydrolyze that beta-1,4 O-glycosidic linkage between glucose units. That's why we can't digest cellulose. Now, I talked about glycosaminoglycans, okay? They're polymers of disaccharide repeating units, which contain either glucosamine or galactosamine that have having acidic sulfate or carboxylate groups, okay? Glycosaminoglycans are found in the extracellular matrix in structures called proteoglycans, which are a complex of carbohydrate and protein, are very large, and play roles in cushioning, lubrication, and adhesion. Proteoglycans do, okay? So let's look at some different repeating units, disaccharide repeating units that characterize different glycosaminoglycans, also called GAGs. So here at this table on the left, we see, you know, the name of the glycosaminoglycan and the approximate number of the disaccharide units that exist per chain. And then on the right, we see a depiction of the disaccharide unit, okay? So hyaluronate, or hyaluronic acid, contains about 50,000 of these disaccharide units, which look like this. Gluconate, through the carboxyl group there, is linked with via a beta-1,3 linkage to N-acetylglucosamine. Okay, and then that's connected to the next disaccharide by a beta-1,4 linkage. Okay, in chondroitin 4 sulfate, this is a much smaller polysaccharide, having only 20 to 60 of these disaccharide units, which contain gluconate, linked via a beta-1,3 linkage to N-acetylgalactosamine, galactosamine, not glucosamine, galact. See how that has been modified on the 4th carbon with the sulfate. See that sulfate there? So instead of an OH of galactose, we've got a sulfate group. And then this disaccharide is connected to the next one by a beta-1,4 linkage. All right. Keratan sulfate is another common glycosaminoglycan. Contains a galactose connected via a beta-1,4 O-glycosidic linkage to N-acetyl-glucosamine that has a sulfate on carbon 6. Okay, and it's connected to the next disaccharide by a beta-1,3 linkage. And then heparin, which has 15 to 90 of these disaccharides. Keratan sulfate had 25 about. What do we see here? A sugar known as iduronic acid, okay, or abbreviated IDO, which has a sulfate on carbon 2. Okay, it's connected through an alpha-1,4 linkage to what is this? Some highly sulfated glycosamine unit. So here's a sulfur here on what? On the amino group itself, see that? There's a sulfate on carbon 3, and there's a sulfate on carbon 6. And it's connected to an alpha-1,4 linkage to the next disaccharide. So these are so-called glycosaminoglycans. And they're found in what are called proteoglycans. So proteoglycans are an example of what are
Dr. Burns?
called the glycoconjugate. Yeah.
Why is there a difference in the linkages? Why is there beta-1,3 and then beta-1,4?
You mean the first two are beta-1,3, this is beta-1,4?
Yes.
Now that is, I'm not sure if you're asking a philosophical question or if you're asking, I don't think you are. I think you're asking, how do I know that this is beta-1,3, right? And how do I know this is beta-1,3?
No.
Is that what you're asking?
No, I'm asking why is it 1,3 instead of 1,4?
I really don't know.
Okay, thank you.
You're welcome. So proteoglycans, right, are one type of glycoconjugate, which also includes glycoproteins and glycolipids, including glycosphingolipids, okay? On the right in this box, you see a key, which is showing all the different kinds of sugars or modified sugars that are present in these various oligosaccharide groups attached, or polysaccharide groups, or oligosaccharides. So that brings me to the difference then, right, between proteoglycans and glycoproteins. So in glycoproteins, we talked about these before. They are proteins that either are secreted or exist on the outside face of a cell, in the extracellular phase, that are modified or decorated with these carbohydrate groups. So here's a glycoprotein on the outside of the cell. It has a transmembrane segment, and then it's got an O-linked glycan structure here, another one here, where this one is linear, that's branched. You see the composition, right? And N-linked glycans here with different kinds of branched oligosaccharide groups, okay? But then in the proteoglycans, you see a much longer polysaccharide component attached. In this case, it's to serine residues through an O-linked glycosylation. I'm not going to talk a lot about glycolipids. What do I want to say here? When you attach, you know, the carbohydrate to the serine, threonine, or asparagine, those residues exist within a certain structural context. Not just any serine, threonine, or asparagine will accept or can be the site of attachment, right? The locus of attachment. Glycosylation is present, again, as I mentioned, secreted proteins such as these different glycoprotein hormones, erythropoietin. This should be LH, not LSH. It should be luteinizing hormone or thyroid stimulating hormone. These are all glycoprotein hormones, or you can also have glycosylation on the extracellular face of proteins or on lysosomal proteins, as we'll discuss shortly. And this glycosylation is important, can be important for the stability and the activity, the bioactivity of the protein. So for example, erythropoietin is a secreted protein hormone that stimulates blood production of red blood cells when oxygen tension is low. We've found a way to produce EPO using Chinese hamster ovary cells, that's a type of mammalian cell, so that we can have faithful glycosylation patterns. But there was a time, and only recently have we gotten to the point where that glycosylation pattern is faithful. And so the EPO we had been producing was not as bioactive, but now we've solved that problem, and it's a lot of it is due to the glycosylation pattern that's present. It needs to be faithful, like the real thing. What else do I want to say? I forgot, but that's it. These are glycoconjugates. Now let's talk a little bit about proteoglycan.
I have a question.
Yeah.
For the slide, you said to change LSH to.
Yeah, LH instead of LSH. That was the typo.
Okay.
It'd be luteinizing hormone, not whatever that is. Now, proteoglycans, okay? Shown here in this figure is actually not just a proteoglycan. It's actually a proteoglycan aggregate, not a proteoglycan per se. Aggrecan, that part of it shown here, is a proteoglycan. It has a protein component and a carbohydrate, polysaccharide, heteropolysaccharide, glycosaminoglycan component. It has, this protein, which has 3 globular domains, G1, G2, and G3, and it's got got keratan sulfate and chondroitin sulfate groups or moieties attached. What else? It has a molecular weight, aggrecan has a molecular weight of around 2,500 kilodaltons, very large. It's a component of cartilage. It plays a role as a shock absorber. And these globular domains are involved in G1 binding to hyaluronate, okay, and also cell adhesion and chondrocyte apoptosis. I suppose the others are involved in that. Okay, but this is an example of a proteoglycan aggregate, and here's another depiction of it. We see the hyaluronic, hyaluronan component, which is it's a glycosaminoglycan with with these aggrecan groups that look like bottle brushes projecting off. And this is an electron micrograph of the whole thing, the aggregate, right? And it plays a role, again, in cushioning and as a shock absorber in the cartilage. Makes sense, right? And then the extracellular matrix of proteins inside tissues contains this proteoglycan component shown here, right? We just saw it, there it is. But also present are cross-linked fibers of collagen, that's type 4 collagen that we saw earlier. And then a protein known as fibronectin, okay? which is part of that whole structure, that matrix. And then fibronectin interacts with integrin molecules, which span the membrane and also interact here with actin filaments inside the cell. Okay, so that's what the extracellular matrix looks like, and proteoglycans are part of that. And here, we're getting close to the end of this, by the way. The bacterial cell wall of gram-positive bacterium, such as Staphylococcus aureus, looks sort of like this, which doesn't really tell us much, does it? This is the cell membrane here. We see the cell wall structure. And if we look within the cell wall structure, we see something that looks like this. What we have is this repeating N-acetylglucosamine and N-acetylmuramic acid units that are connected through what kind of linkages? Beta 1,4 O-glycosidic linkages. So there's this, this carbohydrate, polysaccharide, heteropolysaccharide component of the cell wall. All right. And then we also have these linkers, these peptide linkers that link the heteropolysaccharide chains. And what do we see? We see this pentaglycine bridge connected to this other, what? L-ala-D-glucose, L-lysine, D-alanine, short peptide chain. And so these peptide chains then are important, right? In sort of linking these polysaccharide chains together as part of the entire cell wall structure. Now, we talked about some things, right? Now, I want to mention lysozyme. Lysozyme is an enzyme that hydrolyzes these beta 1,4 linkages between the sugar, the modified sugars of the polysaccharide chains. That's lysozyme. We also talked about penicillin earlier, the antibiotic. It's a suicide inhibitor of a glycopeptide transpeptidase that actually catalyzes the formation of one of these bonds in this peptide linker, that bond between the D-ala and the glycine. That's what the glycopeptide transpeptidase catalyzes. Penicillin inhibits it and prevents the formation of that bond, hence prevents the formation of an intact cell wall structure, allowing the cell to lyse. So actually penicillin is more of a bactericidal agent. We talked a lot about penicillin earlier. We looked at enzymes and so-called irreversible inhibition and suicide inhibition. So penicillin is a suicide inhibitor of the glycopeptide transpeptidase that is important for cell wall structure formation in gram-positive bacteria. Getting close to the end here, folks. Yes, very close to the end. But I wanted to mention that glycoproteins are formed in the endoplasmic reticulum and the Golgi complex. And the Golgi complex serves as the so-called sorting center of the cell. Lots of alliteration there. So proteins that are destined for secretion, the cell surface, or the lysosome are synthesized by ribosomes on the rough ER. You see these ribosomes attached to the outside cytosolic surface. They synthesize proteins which are extruded into the lumen of the ER, okay? Now, glycoproteins that have N-link glycosylation or begin to be made in the ER. So the glycosylation of those proteins, N-linked, begins to occur in the ER and continues to occur in the Golgi complex. O-link glycosylation of proteins occurs in the Golgi, not in the ER. And the Golgi complex, again, is the major sorting center of the cell. The signals as to where the protein should go, okay, is encoded in the amino acid sequence of the protein and its three-dimensional structure, okay? And I want to focus on a particular kind of disease here called eye cell disease, okay? What happens in eye cell disease is that a whole series of enzymes are delivered to the wrong destination in the cell. And the result is that these proteins are found in high levels in the blood and the urine. There's a class of maybe a half dozen hydrolases or hydrolytic enzymes that should normally be trafficked, if you will, to the lysosome, okay? Now, in order for them to go there, they have to have a particular modified sugar unit on them, mannose-6-phosphate, okay, shown here, mannose-6-phosphate, okay? Mannose-6-phosphate is actually made from a larger structure on the protein, and there's a couple of enzymes that are required. In eye cell disease, one of those enzymes is missing. And so you don't get an intact mannose-6-phosphate residue on these hydrolytic glycoproteins that are supposed to go to the lysosome. They don't make it there. They end up, you know, going into the blood and urine instead. And so the lysosome becomes engorged with molecules that should have been degraded, like glycosaminoglycans and glycolipids. This engorgement causes the disease. And it's called eye cell because the lysosomes have inclusion bodies within them, okay? Inclusion bodies, so it's called eye cell disease. Now, all sorts of really bad things happen, okay? There's severe psychomotor retardation and skeletal deformities as a result. The disease is known as mucolipidosis type 2. It's a type of lysosomal storage disease. Certain types of muscular dystrophy, by the way, also result from the improper glycosylation of membrane proteins. So now, you know, this mannose-6-phosphate, this is supposed to be present on these glycoproteins to go to the lysosome. It's not, so they don't make it there, so you have this. And there are different signals, right, for different destinations. Some proteins are glycoprotein hormones that are secreted from secretory granules, right? Others butt off and fuse with the cell membrane to it's vesicles that fuse, and they're located in the membrane. So these are actually transmembrane proteins or intermembrane proteins. Right, Golgi complex is the sorting center of the cell. And then close to the end here, I want to talk about what are called blood types. Okay, blood types. We have type O blood, type A, type B. Each of these different blood types have different so-called blood antigens, O antigen, A or B, that are characterized by the presence of characterized by particular oligosaccharide groups that are present on proteins and lipids on the red blood cell surface, on the blood cell surface, okay? And here's the key for the composition of these oligosaccharides in the O, A, and B antigens. Now, what you notice is that O is kind of like a foundation structure, and A and B are variations of that, where you have in A, right, a N-acetylgalactosamine attached through an alpha-1,3 linkage to a galactose, that galactose in the third position, right? But in the B antigen, we have a galactose attached. So there are going to be different so-called glycosyltransferase enzymes in these different, in people with different blood types that attach either a galactose or an N-acetylgalactosamine or galactose at that position. So slightly different enzymes, slightly different galactosyltransferase enzymes. Now, when people have O-type blood, they don't have a functional galactosyltransferase enzyme of either kind. Nothing is attached, right? Turns out there's an alteration in the gene for that enzyme such that a truncated version of the protein, of the gene is, of the enzyme is produced that's not functional. Now, blood typing is very important for transfusions. If a person is given the wrong blood type, the body will mount an immune response. You'll have red blood cell lysis, and it's not a good situation because the body's immune system will recognize the blood as foreign, right? If you give a person the wrong blood type. The result will be, again, red blood cell lysis, hemolysis, hypertension, kidney failure, shock, even death. Okay. People who have type AB blood are known as universal acceptors. People with O negative in particular are called universal donors. Universal donors. And you can think about why that's the case. Why, you know, someone, for example, with type O blood would be a universal donor. And why someone with type AB could accept any kind of blood. I'll let you think about that. And then finally here, glycan binding proteins. These are cell surface proteins that recognize and bind specific carbohydrate structures on neighboring cell surfaces. Okay. Glycan binding proteins. One class of glycan binding proteins is known as lectins. Examples include concanavalin A and agglutinin. Glycan binding proteins, lectins in particular, mediate cell-cell recognition, cell adhesion, cell signaling, and protein targeting within the cell. Now that last thing there, cell signaling and protein targeting within the cell, that's what mannose-6-phosphate mediates. It's a lectin. Wait, let me say that differently. It's not quite true. The mannose-6-phosphate receptor, which recognizes mannose-6-phosphate on those hydrolytic enzymes, is an example of a lectin, right? The mannose-6-phosphate receptor. And it helps guide the protein to where it needs to go in the lysosome. So each lectin protein contains 2 or more sites for binding carbohydrate units. The interactions act as a molecular Velcro. And I'm here talking about lectins on the outside surfaces of cells. These interactions allow cell-cell adhesion, so it's sort of like molecular Velcro, where each lectin protein contains 2 or more sites for binding carbohydrate units. Acts as a molecular Velcro, allowing the cells to adhere to each other in a specific fashion through that recognition between the lectin on one cell and the carbohydrate groups on the other. Now, I mentioned this mannose-6-phosphate here just now. Hemagglutinin is an influenza virus protein that recognizes sialic acid residues that are linked to galactose on cell surface glycoproteins. And it's the site of viral particle entry. By the way, the flu vaccine is going to be, should be available soon for people to have if they, scientists and health officials are encouraging people to get that vaccine this year, flu, so that, you know, as we deal with the coronavirus pandemic. And finally, merozoites of the malarial parasite Plasmodium falciparum gain entry into red blood cells via their interaction with a glycan binding protein and the erythrocyte surface glycoprotein, glycophorin. So you know how we talked about glycophorin earlier as an example of an integral membrane protein? Well, that's actually how Plasmodium falciparum gains entry. It has a glycan binding protein on the surface of merozoites, which is a form of the parasite, allows entry into red blood cells. So they, you know, that's an interesting example of a glycan binding protein. Hemagglutinin is another example. Okay, are there any questions? Any questions about carbohydrates? So what I see is that I just barely finished this lecture in 2 hours. So I didn't get to cover glycolysis at all, did I? I guess that's why I had originally had it to be a flipped classroom discussion and had you view it before class. What happened was we had a restructuring of the course materials, and I was short a lecture as a result, and so I moved that to a flipped class, but somehow the link to the recording didn't make it this year. So what we'll do is we'll just try to catch up as we go along. The TCA cycle discussion is about an hour long, so my guess is after next period we'll be about an hour behind, and we'll just try to make up as we go along. So that's it. Do you have any questions? Any questions? I should be able to give you guys an update and give you an answer on your exam by this evening. But anyway, I hope you have a nice rest of the day, and I will see you on Thursday. Thank you, Dr. Sutherland, for being a co-host and recording the lecture.
I'm now stopping recording.
Thank you. Okay, see you guys on Thursday morning.